Hello class, today we're going to start lecture 2 of unit 3 where we're going to be discussing carbohydrate digestion and microbial fermentation. We're first going to focus on non-ruminants because even ruminants have these same components that non-ruminants do. So we'll start off with the non-ruminants. So all animals end up breaking down polysaccharides and disaccharides, and we use 1 to 3 steps. First, we have salivary amylase and pancreatic amylase that break down starch into smaller polysaccharides. So our salivary amylase and our pancreatic amylase, both of these are alpha amylases. And you can see here that they are, we have, in this picture we're just showing pancreatic amylase, but we have the pancreatic amylase is going and breaking down this starch granule, and we are getting amylose and amylopectin, and then the end products end up being maltose, maltotriose, and limit dextrins. Now, our maltose, our maltotriose, and our limit dextrins, especially our maltose, that is a disaccharide. So first you can see that these disaccharides, they have to get through this unstirred water layer. So this is the same as the mucus layer that's covering our enterocytes. And once it diffuses through this mucus layer, you have the maltose or the maltase enzyme that is right here. Our maltase enzyme is able to break down maltose and the maltotriose, and we get glucose. And our glucose enters in with sodium through SGLT1 into the cell, and then that's able to then go to the basolateral membrane and enter the blood. So we have our disaccharidases. So maltase is the main one when we're discussing starch specifically, but there are many other different types of polysaccharides and disaccharides. So we're going to have maltase, sucrase, lactase, all are constitutively expressed on the microvilli. So when I say constitutively expressed right here, that means that they are always expressed. They're not upregulated or downregulated based off of how much you've eaten. They're always on the microvilli. Compared to the pancreatic enzymes, those are being released based off of what you have just eaten. And then once the sugars are converted to monosaccharides, they are then absorbed by the enterocyte, and we have 2 main ones that we're going to be focusing on: SGLT1, which we've already talked about, which is sodium glucose transporter 1, and this does glucose as we mentioned before, but also what we didn't mention in the last unit was it absorbs galactose. So it has a higher affinity for glucose, so it prefers glucose, but galactose and glucose are pretty similar, and so they are going to have a similar, be able to share the transporter. Now, fructose is completely different than glucose and galactose, so it utilizes and is absorbed through GLUT5. So GLUT5 is a little different. It requires no energy to be absorbed. So if you remember our SGLT1, although it isn't specifically using energy to get the glucose and galactose in, secondarily, it needs the sodium-potassium pump that utilizes 1 ATP to get the glucose into the cell and the sodium out. Now, interesting, I found this study which showed that the amount of salivary amylase activity a person has, it ends up predicting the diabetes. So in this study, they took people and they swabbed their mouth, collected their salivary amylase, and classified them either as low activity or high activity. And then they gave those individuals a starch bolus. So they were given a pill that had a certain amount of amylose in it, and had them consume that product. And over a period of 2 hours, they looked at not only the plasma glucose, but they also looked at the insulin value, and both of those results were highly correlated. Here I'm just showing you the graph of the plasma insulin. So one of the interesting things was with these groups, you could see that based off of the diagram here, that those that had low activity, which is these dark circles here, they, after about 30 minutes, they had much greater insulin levels in their plasma. In addition, they had greater glucose levels in their plasma if they had low amylase activity. However, if they had high amylase activity from their saliva, they had much lower levels of both plasma and insulin. And this only happened when they were provided starch. They gave these individuals also, they did a glucose tolerance test where it was just glucose, and there was no difference in the amount of plasma glucose or insulin between these two groups if it was just glucose. So therefore, the authors ended up concluding that these healthy individuals that had low salivary amylase must have a greater risk for diabetes. So they had greater blood glucose and greater insulin, which if you're always having higher blood glucose and you're always having higher insulin, you're more at risk at becoming insulin resistant. So your cells may not respond as quickly, especially if you end up being on the heavier side. So this would end up being type 2 diabetes that these individuals are more prone to. So as I mentioned, with amylase, we have two different types. We have salivary amylase, and overall salivary amylase ends up resulting in very little starch digestion. If you were to take a saltine cracker and put it in your mouth and let it sit there for about a minute or two and just keep chewing it, you will notice that saltine cracker over time gets sweeter and sweeter, and that's due to your salivary amylase. And I'm sure many of you know that when you're eating, it's not like you're going to be having food sit in your mouth for an extended period of time. So really, there's not that much time for the salivary amylase to work. And once it hits the stomach, it ends up becoming denatured. So the optimal pH for salivary amylase is around 7 to 8. So once it hits that low pH of 2, it stops working. In addition, so humans are able to secrete salivary amylase, but there are many animals that do not secrete any salivary amylase. So this includes the dog, the cat, the horse, the cow, sheep, and goats. So none of these animals secrete salivary amylase. So the main amylase that we focus on, that is doing the predominant amount of work, is pancreatic amylase. And both, as I mentioned before, both salivary amylase and pancreatic amylase are alpha amylase, but pancreatic amylase is the predominant starch enzyme. And there are basically two types of pancreatic amylase that end up working. One ends up cleaving at the alpha-1,4 in a random fashion. So it goes along the amylose and just randomly cleaves throughout the polysaccharide. So it will go in the middle, and then we'll get two shorter but still long polymers of amylose. And then we have a second type that only works at the end. So it ends up removing maltoses from the end. And the amylases end up, so if we had our glucose molecules, so if we have our glucose, chain here, bear with me while I'm trying to draw. Oops, I forgot the V. So we have our amylase will actually go in here, and if this is 1, 2, 3, 4, 5, it cleaves in between the third and the fourth glucose unit. So that's where our pancreatic amylase is breaking. Now, with our so once we have our polymer chains of carbohydrates broken down to disaccharides, we have our disaccharadases that are able to work. And interestingly, there is the effectiveness of the disaccharadases actually ends up depending on the maturity of the GI tract. So here, for instance, this is some data looking at the maturity of the GI tract of piglets. So you could see, and if you are unfamiliar with piglets, when piglets are first weaned, they're weaned between 18 and 21 days of age. So they're still very young. Naturally, they're not going to be weaned that young by their mother, but for commercial purposes, we wean them younger. And then we put them on a weanling diet. And those weanling diets, typically we end up putting lactose in those diets, or whey, which has lactose in it. And the animals actually have a requirement for lactose, and that's all because they do not yet have the maltase enzyme well developed, or the sucrase enzyme well developed on their GI tract, but they have a significant amount of lactase still there. So here we have, looking at the absorption of different of both glucose and galactose absorption in these piglets, and we put them into two different groups. One group was given lactose is the main energy. So here we have lactose, and the other one was given maltodextrin, which is basically maltose. So you can see that the animals that were provided lactose, they right when they were given the diet right away, they have a significant amount of absorption in both glucose and galactose. And that's because lactose is made up of glucose and galactose. So we're going to have a significant amount of glucose and galactose, and that's all due to the lactase enzyme that is functioning. However, when the piglets were providing a maltodextrin diet, that we really did not have a significant change in the net amount of glucose or galactose that was absorbed. So basically, how does that end up affecting the animal? Usually these piglets, if they are not provided a diet that has lactose, if they were put on a more mature pig's diet, they're going to end up getting a lot of diarrhea. You end up having many changes within the GI tract. So here in graph B, you could see the cumulative hexose absorption. So in the lactose diet, we have a significant amount of glucose and galactose, much greater amounts of those absorbed over the whole study compared to the animals that were provided the maltodextrin and the maltodextrin diets, which had glucose, and obviously there's no galactose in maltodextrin. And so in C, we have the different components of looking at the lactose diet, how much everything was absorbed, and in which components. And you can see with the lactose, we had much more of it absorbed than those with the maltodextrin. Now, also interesting, if you were to look at the health of the GI tract. So here we have in A, in this picture, you see these in A, these are the piglets that were provided the lactose diet, and we have really nice, pretty, long villi, very healthy, a lot of surface area. And compared that to the animals that were provided the maltodextrin, their villi are much smaller, they're not as healthy, and that's all because we've now, it's very difficult for them to absorb that. We have a lot of microbes that are needed to break that down, so it's much more complicated for the animal. So this is just a summary of the carbohydrate digesting enzyme. So for the mouth, we have salivary amylase, and related to carbohydrate digestion, we don't really have any digesting enzymes that are being secreted. Our amylase is also coming from our pancreas. And then all of our disaccharidases, including trehalase, lactase, maltase, and sucrase, those are all constitutively expressed on the enterocytes. Now, we talked a little bit in the last lecture about cellulose and how it's different from starch, and cellulose is the most prominent carbohydrate in the world, and the second most prominent carbohydrate in the world is chitin. And chitin, if you remember, is what makes up the exoskeleton for insects and crustaceans. So if you like to eat crab or lobster, that outer shell is all made up of chitin. So this is chitin is a linear polymer of N-acetylglucosamine, and you can see we have the glucosamine, the acetyl groups that are conjugated to the glucosamine, and these are beta-1,4 polymers. And as I mentioned, it's the second most abundant. So a lot of animals, especially our obligate insectivores, need to be able to break down this chitin. One, there's a lot of energy potential there. And two, we need to be able to break that so we can get to the nutrients inside. So there are two enzymes. So a lot of animals, especially our obligate insectivores, have these enzymes that are necessary to digest chitin. So it requires two enzymes. The first one is chitinase, and this is released from either the gastric or pancreatic juice. And this all depends on the animals. Some animals only have it secreted in the stomachs, and others have it secreted from both the stomachs and the pancreas. And really it depends on, are they obligate insectivores or are they omnivorous that also consumes insects. So our chitinase is what's going to yield dimers and trimers. So it's similar to what our amylase is doing. So we're getting small dimers. We're getting 2 or 3 of the glucosamine, acetylglucosamine conjugated together. So that's coming from the stomach or the pancreatic juice. Now, once we have those dimers and trimers, we also have chitobiose, or biase, sorry, that's supposed to be biase. And so this is just like maltase, sucrase, and lactase. It's constitutively expressed on the microvilli. And this is what is breaking up those dimers and trimers, and it's yielding monomers of the acetylglucosamine. So for the exam, I want you to be able to tell me that chitinase is breaking it down from the stomach, released from the stomach or the pancreas, depending on the species, and the chitobiase is constitutively expressed on the microvilli. And this could be some potential where, if we think of a way to apply, if you were to find an animal and you didn't know what it ate, one thing you could do would be see if it expressed a high amount of chitinase or chitobiase. Would be a potential exam, higher order thinking question that may come up. So this is just a table showing the chitinase activity of different species and which ones are having the chitinase released from the gastric mucosa or from the pancreas. So if you notice that the majority of the amphibians and reptiles that are obligate insectivores, they are releasing the chitinase from both the stomach and the pancreas. And we do have a couple, like the chicken here, it has the chitinase released from the stomach, from the gastric mucosa. So it's the chickens are omnivorous. They're not obligate insectivores, so they don't really need it in the pancreas. Compare that to another avian species, the starling, which has both the gastric mucosa and the pancreas having the chitinase. Now, interestingly, some people actually do have chitinase activity. One study I found was done by an Italian group where they took 25 people and looked at the activity or the expression of chitinase and from the gastric region. So these were individuals that were actually going in for some sort of surgery, and so they were able to take biopsies of the stomach, and they looked for expression of chitinase. And out of the 25 people, they had approximately 12 that did have expression of chitinase. Now, interesting, this was a group of Italians that haven't, insects are not a major component of their diet. So it would be interesting to have a similar study done from groups of individuals that utilize insects as a major food source. And maybe they have a greater amount of chitinase that is expressed. And also, fun fact, anybody that wants to increase the amount of insects that you consume, you can actually go and buy like various insect meals and use that to bake. So if you ever get the itch to increase your protein level and want to use insects, you can actually buy insect meal and replace that in your food. So once we have our monosaccharides, we need to absorb them. And we've already talked about SGLT1 and sodium potassium pumps, so we're not going to spend too much time on this, but it's always good to review. So our SGLT1, remember, SGLT1 specifically does not require energy. So it does not have an energy requirement. However, indirectly it does. So SGLT1, it's driven by electrochemical gradient, which is maintained by the sodium potassium pump. So we have one glucose and two sodiums. If you remember, sodiums bind first, That causes a conformational change. Then we have one glucose that binds. That causes a conformational change. And both of these things are able to enter. Our glucose is then able to either be metabolized by the enterocyte or leave into the blood through GLUT2, where now we have this high level of sodium found within the blood. And we need to kick this sodium out so we have more glucose that's able to be absorbed. So that sodium, and it's going against its chemical gradient, so we use sodium-potassium pump and we kick that out. And we end up bringing in 2 potassiums, and we use 1 ATP. So indirectly, SGLT1, the absorption of glucose, indirectly is utilizing energy. And this also, glucose or galactose, it just preferentially, glucose has a higher affinity, but galactose is the same. And so also know for here that we're going to be kicking out 3 sodiums and bringing in 2 potassiums. So we actually are getting rid of an extra sodium, so we maintain that level of sodium to be really low so we can really encourage the glucose and sodium to come back in. So here's a little video of the sodium-potassium pump.
Energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
So there you go. That was a pretty decent video of summarizing the sodium-potassium pump and how it works. So as I mentioned before, we have other glucose transporters that are utilized. So SGLT1 is what glucose is coming in and galactose are coming in through, but we have other ones that are expressed all throughout the body. So one of the first ones that was discovered was actually GLUT1. So GLUT1 is a glucose and galactose transporter, and it is expressed constitutively on our fetal tissue, our red blood cells, and our blood-brain barrier. So now my question is why? Why are these ones always have GLUT1? Well, if you think about it and you remember what I mentioned before with the exclusion principle, is that these are the tissues that have to use glucose. So our brain has to use glucose. Unless you're in an extended starvation state, your brain is going to be using glucose to meet its energy needs. In addition, our red blood cells have to have glucose. Remember, they don't have mitochondria, so they're mainly going to be utilizing glycolysis and converting the glucose to pyruvate. Pyruvate then leaves the red blood cell, or it's converted to lactate. Lactate leaves the red blood cell, and that's the Cori cycle. Then we have our fetus. Our fetal tissue also is always expressing GLUT1 because our fetus only uses glucose for a energy source. And then if you remember with our GLUT2, this is the transporter for glucose, galactose, and fructose to leave the enterocytes. It's also expressed on the hepatocytes, renal cells, and our beta cells. So can anybody think of where our beta cells are located? So these beta cells are what are making up the islets of Langerhans. So these are found within your pancreas. Sorry about my poor handwriting. So those are how the glucose, galactose, and fructose are all coming in and out of the cells. And then we have GLUT4, and this is the one that the majority of people think of when they're thinking about glucose. Because GLUT4 is insulin dependent. It's an insulin dependent transporter, and it is found on adipose and skeletal muscle. So basically, when we have our insulin bind the insulin receptor, that causes signaling in the cell. You have second messenger signaling that then causes the glucose transporter 4 to be moved up onto the outside of the cell membrane, and then glucose can transfer in. So our adipose tissue and our skeletal muscle is the lowest on the totem pole, so they get the scraps of the amount of glucose. So if there is not adequate glucose, then they are going to have to change to a different energy source. So they're going to have to utilize either our fatty acids or ketone bodies as their energy. And then we have, as I mentioned before, GLUT5, and this is our fructose transporter. And it is on our enterocytes, and it's also found on sperm. So how does carbohydrate digestion differ with herbivores? And basically, not much. The host animal, when they have glucose or monosaccharides to absorb, if it gets to the small intestine, everything is going to be the same as our non-ruminants. The biggest difference is with our obligate herbivores. They heavily rely on microbial digestion. So our ruminants are utilizing the forestomach, and our hindgut fermenters are obviously using the hindgut. And the main products are microbial protein, and we also have the volatile fatty acids, being the acetate, the propionate, and a butyrate. And then we have gases that are formed. So we have methane gas, carbon dioxide, and hydrogen. All of these are produced anaerobically, which means that there is no oxygen around. So we've seen this picture before. This is summarizing our anaerobic fermentation. So we have, regardless of what the carbohydrate polymer is, if it's cellulose, hemicellulose, pectin, fructans, or starch, those all come in, and depending on the microbes and their metabolism, they're all basically going to be converted to glucose. We're going to get single things of glucose. And then all of those are converted to glucose. Our glucose is then converted to pyruvate here. And you can see then we, depending on the species of microbes found within the GI tract, we are going to get different products. So here we have our acetate at the bottom, our butyrate and our propionate, our volatile fatty acids. We also have our methane gas. And depending on the species, some of the bacteria, especially if we have a lactobacillus or whatnot, they're going to be able to convert pyruvate straight to lactate or lactic acid. And notice that all of the products of microbial fermentation is they all are acceptors of electrons. So our microbial digestion of carbohydrates varies with the species of animal. In addition, it varies with the species of microbe that are within the GI tract. So you could have two different horses and they're going to have different microbes, so therefore they may have two different types of microbes found within their GI tract. And a lot of that depends on what the animal is fed. Similarly, especially if we're talking about ruminants, if you take a grass-fed beef cow compared to a feedlot finished steer, we're going to have different species of microbes found within the gut based off of what the carbohydrate that the animal was eating. And the concentration of the volatile fatty acids varies with the site of microbial digestion. So our foregut fermenters are getting much greater amounts of volatile fatty acids produced compared to our hindguts. Our hindgut fermenters are not as efficient at absorbing all the volatile fatty acids that are produced. And then we have our pH inversely varies with the volatile fatty acid concentration. So that all depends on which VFA is the predominant one. So if we have predominantly acetate, we're going to have a higher pH. If we have predominantly lactate, we're going to have a lower pH. So here we have a graph showing the variation of volatile fatty acid production in different animals. So if you notice that these are all either simple stomach monogastric animals or they are hindgut fermenters. Ruminants are not included in this particular study. So you notice, and these are on the x-axis, we have different sections of the GI tract. So we have this is section 1, 2. Oh, this is stomach 1 and stomach 2. The small intestine, so duodenum, jejunum, ileum. This is the cecum, and colon 1, colon 2, and colon 3 is what those mean. So our GI concentrations varies by the site and extent of microbial fermentation. So you can see here that the pig, and especially if you remember the anatomy of the pig, they have a lot of volatile fatty acid production within the cecum. And they also have a pretty extensive hindgut colon. So they still have a lot of fermentation that's occurring in their colon. Compare that with, let's say, the dog. The dog, if you remember, has a very small cecum, so there's not very much fermentation that's occurring there. Where we have, it's mainly in the distal colon where they're having the majority of their volatile fatty acids that are produced. Our pony, also another one, very high amount of VFAs produced within the cecum and the first portion of the colon, where the latter portions of the colon, very small amounts of VFAs that are produced. So how does it differ with ruminant species? So this is showing the relationship between the volatile fatty acids concentration and the pH of the rumen. So here, if we are looking at a pH 7, which this is going to be a hay-fed animal. So our hay-fed animal, pH 7, we're going to have predominantly acetic acid and propionic acid. And then a little bit down here, we would have butyrate. So let's say these are, we have some, so this would be our grass-fed beef. Now, as we change those animals over, and let's say we put them over to a feedlot, so our feedlot animals are typically their diets contain about 60% concentrate, which are going to be more grains. So they have a lot of ground corn and steam rolled, etc. So the more concentrate diets that we get, we're going to end up slowly decreasing the acetic acid. And as we decrease the amount of acetic acid, we end up increasing the amount of propionic acid. So these two are working in conjunction. So if we have less acetic acid, we're increasing our propionic acid. So our ratio, which we want to be 3-to-1, ends up being reduced. So it gets more closely. So at this portion, we have a 1-to-1. And once we get past that, then we start having this lactic acid that's produced. So our pyruvate is then being converted straight to lactic acid. And once we have lactic acid produced, that is greatly reducing our pH. The more lactic acid, the lower our pH. So this table is just showing how the amount of VFAs that are produced are meeting the animal's energy requirements. So obviously we don't have all the details to many of these different species. We can see how much they end up, the concentration of the volatile fatty acids, but we don't necessarily know what percentage of the energy requirement it is meeting. So here we have with our cattle and sheep, and they're going to range between 42 and 79. So our cattle are going to be more in the 65 to 75%. Here, if we look at our wildebeest, so this was found that our wildebeest, about 67% of the energy requirement is met through the volatile fatty acid. So a pretty significant amount of our energy requirements in our foregut fermenters are being met with the volatile fatty acids. Contrast that to the hindgut fermenters. So here we have the amount of the VFAs that are produced, and you can see the dog, and if you remember the GI tract of the dog, we only have about 2% of the volatile fatty acids of the dog are meeting their energy requirements. And coming up pretty close is the humans. You and I, we are not very efficient at our volatile fatty acid absorption. So our colon, our hindgut is just not long enough. So we can only meet about 6 to 9% of our energy requirements through the assistance of microbial fermentation. So if you know anybody that loves their green leafy vegetables, and that's the main thing that they are consuming to meet their energy requirements, the reason why they're so skinny is because we have they're not able to absorb all of those VFAs. And then you have our Our pony here, we had 46%. They're about the best out of the obligate hindgut fermenters. And the rabbit, pretty similar, 30 to 38%. And then our pig is about 10 to 31%. So what are the factors that are affecting microbial fermentation? So the composition of the diet, and that is best described utilizing the ruminant. So if you see how the grass-fed animals, what they're fed, and how that's affecting their pH, so they have much higher pH, and that's because of the microbes that are able to survive within that environment and break down that grass compared to a steer that's in a feedlot given a concentrate diet. We also have the nutrient load. So how nutritious is the food or feed that the animal is consuming. And then we have the amount of water and digesta flow through the compartments is also altering the microbial fermentation. So here's another graph showing more of with cattle specifically. So here we had some, I believe it was dairy cattle, and we can see how the concentration of fiber altered the ruminal pH. So we had the cows were fed either a hay diet, coarse grain, so it's going to be corn that's just rolled corn or rolled oats, and then we have fine grain. So these are going to be finely ground and then incorporated into the diet. So the cattle that were provided the hay, they always have a pH greater than 7 throughout this whole thing. And our coarse grain, because the ruminant microorganisms still have to break up some of that cell wall for the animal, their pH starts at 7 and then goes down and it eventually comes back. But our fine grain, they start at a pH less than 7. It drops down to about 5.5 and eventually comes back. So the greater the energy density here, so this is going to be our fine grain. So our fine grain is going to have higher energy density than our hay or our coarse grain. So that's going to decrease our ruminal pH. So why is that happening? Well, our higher energy density ends up also having a lower amount of fiber. So the lower amount of fiber we have, we end up decreasing the amount of saliva. So why is that affecting the pH in the rumen? Well, if you remember, especially with ruminants that they have the majority of their saliva is coming from the parotid salivary gland, and that salivary gland is providing a serous saliva, which is mainly made up of water and buffers. So if we have less saliva, we end up having fewer buffers. We end up having less water flow because we're decreased the amount of saliva, and that serous saliva is mainly water. And that ends up resulting in we have less ruminal contractions. We have less ruminating that is occurring. So they also don't have to chew the cud or regurgitate and chew and have more saliva produced. And then that ends up altering the VFA production, where we get a significant amount of lactic acid produced. And you can see that after the animal eats, about 2 hours after eating, we have that the pH goes back to where its baseline was. So obviously these animals had been eaten a fine grain diet for a significant amount of time. If you would have taken these animals and provided them a fine grain, we may have had much greater changes. And so why does this pH normalize after 2 hours? And that's because the volatile fatty acids have been absorbed. So we've now gotten rid of all the volatile fatty acids, so it increases the pH. Now, the type of fiber is going to change the ability for the microbes to ferment it. So we have a list of various dietary fibers. Here we have cellulose. You can see that fermentability of the cellulose ranges between 20 and 80%. And a lot of that has to depend on the maturity of the animal, because if you have a more, not of the animal, of the plant, if you have a more mature plant, the cellulose is going to start getting surrounded by lignin. So this drastic difference is due to lignin concentration. So the greater the lignin, the less fermentability of the cellulose. So if you had, for instance, tree bark, you're going to have about 20% compared to freshly cut alfalfa, which is going to be around 80%. Our hemicelluloses are between 60 and 90%. And a lot of this has to do with the fact that some of the hemicelluloses are soluble, some are insoluble. Pectins, it's a soluble fiber, 100% fermentable. Guar gum, soluble fiber, 100% fermentable. Our resistant starch, so this is starch that basically once you have, so resistant starch forms when you, for instance, boil potatoes. So you boil potatoes and you have that water molecule is able to get inside in between those starch components. If you let those potatoes cool, let's say you make potato salad, you boil all the potatoes, you then rinse them, you put them in the refrigerator, and you finish making the potato salad the next day. That has caused resistant starch. So once we have the starch is boiled, if we were to eat that boiled potato, we would be able to absorb all of it. But once it's cooled, that the reaction with the water and the starch, that is what forms what we refer to as resistant starch. So this resistant starch we are unable to absorb, but our microbes can 100% ferment that. And then you have inulin and other oligosaccharides that they're also 100% fermentable. So this is kind of going off of our soluble fibers and our fermentability. So our soluble carbohydrates, our pectins, our guar gums, our soluble hemicellulose are quickly fermented after you eat. So here we have this graph on the right hand side, our soluble carbohydrates. So this would be starch. So when I say soluble carbohydrates, where I'm meaning starch, specifically amylose. Soluble carbohydrates, you could see right away while the animal is eating, basically as soon as it's done eating, that starch has all been fermented by the microbes. Compare that with our pectin. So our pectin isn't as quickly fermented but it's pretty close to our starch. So our soluble fibers and pectin, guar gum, they're all pretty similar. You could see about 3 hours after we peak, and they're all fermented about 12 hours later. Compare that with our cellulose. So our cellulose, it takes probably about 3 hours. Here we start being able to digest it. So over here at this period, that was the cellulose from the last feeding. So while we're eating and we're having more stuff come in, we're still finishing fermenting our cellulose from the last meal. So here it starts about 3 hours into while they're eating, and it comes up, and cellulose is the one that takes the longest to ferment. So there basically is a negative correlation between the rumen pH and the nutrient fermentability. So our soluble carbohydrates and our pectins, our soluble fibers are all, these two are going to end up resulting in a low pH in the rumen, where our cellulose is going to end up giving us a higher pH in the rumen. And then we have a positive correlation between the volatile fatty acid concentration and the nutrient fermentability, meaning that if you remember previously with our starches, or if our concentrate diets, our feedlot cattle, those volatile fatty acids, the reason why we do it, even though it's not all that great for the animal, because they end up getting so much more energy out of it. So they produce many more volatile fatty acids. That volatile fatty acid concentration is greater. So the more soluble the carbohydrate, the greater amount of volatile fatty acids that are produced. So our volatile fatty acids, we talked about this previously, they are absorbed passively, and it involves transport of carbon dioxide. And depending on the specific type of ruminant, we have between 70 and 75% of the volatile fatty acids are absorbed from the rumen. So we're missing about 25 to 30%, but that's pretty good compared to the other animal species. So this is just, again, we talked about this in the last unit, how our VFAs are transported. So you can see here that they are passively transported. So here we have our charged acetate. It can be converted within the rumen to, this is acetate, this is acetic acid. It could be converted, acetic acid can be converted to acetate, which passes into the cell, and that can pass straight out into the blood. If we have acetic acid absorbed, we need to do a little bit of stuff. So if we have acetic acid absorbed, we end up needing to alter it and neutralize it. So with that, we end up having our carbon dioxide. We pull in from the rumen, we add that with water, we get carbonic acid produced. We break that carbonic acid with our carbonic anhydrase, and we release a bicarbonate, and this bicarbonate ends up leaving back into the rumen in exchange for this acetic acid. We take that hydrogen, add it onto the acetic acid, and then it's able to go out into the blood.